In the Bible, the book of Genesis states that God created plants, animals, and humans. Traditionally, the ability to design and produce creatures has been considered to be divine power. Besides the Bible, there are multiple versions of mythological fictions around the world about the origin of creatures. Today, however, Homo sapiens are acquiring this divine ability. It has been 3.8 billion years since the first organisms appeared on Earth, and all of life has evolved following the principle of natural selection. But for the first time in the history of biology, we are going to add a new rule to the game of life. With the exponential progress of gene editing technologies in recent years, including CRISPR-Cas9, humanity is becoming able to modify the blueprint of life. This technology has the potential to eliminate genetic ailments, increase agricultural production, and even upgrade human capabilities. What kind of future will be brought by genome editing? Geneticists had been longing for decades for a technology to precisely target and edit specific genomic sequences. In 2012, a paper on the defense system of microbes called CRISPR-Cas9 was published in Science magazine. It immediately sparked the imagination of biologists around the world. Some bacterial species have evolved a system to cut off the genomes of invading viruses to kill them. The system recognizes the offender viruses by their DNA sequence. The cuts are not delivered at random places, but at specific targeted sites in the virus's DNA. Researchers found that this defense system consists of two critical components, seeker and cutter. The seeker is an RNA encoded in the bacterial genome that looks for the DNA sequence that matches itself in the virus's genome. The cutter is a protein named Cas9. Once the seeker recognizes the virus as an enemy, Cas9 is deployed to cut off the DNA at the spot specified by the seeker. To put it simply, the seeker finds the target spot and brings the cutter to the spot, and the cutter snips the targeted DNA. Researchers discovered that we can edit genes with pinpoint accuracy by introducing two manipulations in this mechanism. First, we can change the target DNA sequence by replacing the seeker. Second, we can interfere in the recovery process of DNA. When DNA is cut open, it tries to recover the lost part, usually from the other copy of the gene in the cell. But if a cell is flooded with foreign DNA, then the gene accidentally copies the information from this external DNA, rather than from its backup. This works more easily, precisely, and efficiently than any other gene editing methods ever existed. With the appearance of CRISPR technologies, we can edit genes precisely and efficiently with a lower cost. In principle, a single letter of human DNA can be mutated to another letter, leaving the 3.2 billion other bases of the genome largely untouched. The cost and efficiency are also important, as research funds and time are obviously limited. Since this new genome editing technology was unveiled in 2012, its various potential applications have been suggested. One notable application is crop improvement. For instance, crops like high-yield rice, non-toxic potatoes, and less perishable tomatoes have already been created using CRISPR-Cas9. Given the climate change and food shortages caused by overpopulation, genome editing for crop enhancement is going to be essential. Also, genome editing can solve the problem of food allergies. Most allergens are specific proteins. We can reduce allergens in food by removing genes associated with the production of allergenic proteins. There are already ongoing studies on genome editing in livestock animals, such as chickens that lay eggs with less allergen. Another application of genome editing is, of course, in medicine. Genetic disorders like hemophilia and muscular dystrophy are caused by mutations in specific genes. Hemophilia, for instance, is a condition where liver cells can't produce blood clotting factors properly and, thus bleeding can't easily stop. By employing CRISPR technology, we can cut off the malfunctioning genes in liver cells and insert the correct DNA sequence. There are various technical challenges that need to be solved, including off-target effects, in which the tool falsely targets wrong sites and delivers unintended modifications. We are not there yet, but before long, we will gain the ability to purge thousands of diseases from our lives. While genome editing offers numerous potential benefits, it presents particularly significant ethical challenges. One of the biggest concerns is that this technology could be used not only to cure disease but also to enhance human capabilities. There is no clear line that separates healing from enhancing. In most cases, medicine is first developed and approved to save people from falling below what is considered to be the standard. However, the same tools can then be used to surpass the standard. For example, plastic surgery was first developed during the First World War to treat soldiers whose faces got injured in battles. When the war was over, surgeons realized that the same treatments could also turn healthy individuals more beautiful. Today, plastic or cosmetic surgeons earn millions by upgrading the wealthy, and we take it for granted. Genome editing might follow the same path. It will begin with parents who hope to eliminate fatal genetic ailments from their babies. But once it becomes possible to edit human DNA to replace deadly genes, we might start using the same mechanism to fix less fatal genes, such as ones responsible for autism and obesity. Who would like their child to suffer from any of these? Furthermore, if you are about to provide your child such treatments, wouldn't you want to give them a little more push by enhancing their memory, athletic ability, or immune system? Even if you are personally against such upgradings, what if the neighbors are doing it for their children? Would you dare to have your child lag behind them? Any upgradings are initially justified as healing. But once it is approved, it may end up being unstoppable by moral discussions. The caste system in Hinduism has divided people into 4 groups based on the family they were born into. They believe that individuals in higher caste groups were intrinsically superior, but this was pure fiction. There is no biological difference between Brahmins and Shudras, Kshatriyas and Vaishyas. Brahmins insisted that they were naturally smarter than everyone else, but even after thorough examination of their DNA, we can never find any sequence unique to Brahmins. Historically, social and economic disparities have not arisen from genetic differences. Rather, they have been the result of cultural and environmental factors, which have been justified and amplified by fictions. However, with the advancement of genome editing, the nature of inequality may fundamentally change. Once it becomes possible to upgrade Homo sapiens through genome editing, we will see real gaps in physical and cognitive abilities between an enhanced upper class and the rest of society. These upgraded superhumans will enjoy unprecedented health, abilities, and creativity, which will further accelerate inequalities. After hearing all of these, your reaction might be to hope that somebody will hit the brakes to stop it, but we may not be able to halt its progress. In November 2018, a Chinese scientist had claimed to have created the world's first genome-edited babies. He used the CRISPR-Cas9 technology to modify the DNA of twin girls, making them resistant to HIV infection. Many scientists and organizations criticized the experiment for its potential impacts and violation of ethical guidelines. However, different nations have different moral codes. If some countries use genome editing to produce geniuses that far outperform other citizens, whose country forbids genetic engineering, will we know when to hit the brakes?